excited to introduce my friend and colleague, Megan Vahsen. Be quick to not take too much time on a brief bio. So, did her BS at College of William and Mary, then a master's at Colorado State University. That's where we met. We didn't really run around in the same circles, but now we're on an online collaborative research writing group that started during COVID. Megan just finished up her PhD at Notre Dame and is now a postdoc at Utah State. And I will kind of brief there and let her go ahead and get started and hand it over to her to talk about her research that she's been doing sort of locally in our area of wetlands.
So thanks so much, Travis, for having me here. I'm really excited to be back in Maryland. I did all of my PhD research at the Smithsonian Environmental Research Center in Edgewater, Maryland. So I've been gone for a little bit. Feels really good to be back. So broadly, who am I? I'm going to talk a lot about marshes today, but I'm really motivated by interesting questions about ecoevolutionary dynamics. So I investigate how evolutionary and ecological processes interact and feedback on each other, scaling from genotypes to ecosystems. I'm going to have some figures that look like this in my talk today. This is what I call an ecoevolutionary feedback loop, where processes that are happening that are generally in evolutionary biology, like selection, adaptation, evolution, can impact what's going on in ecosystem ecology and can feed back on the impacts of climate change. A little bit more context. So I am going to talk about my dissertation research today, but the work that I did at Colorado State was looking at ecoevolutionary dynamics, but with a totally different system, tribolium beetles in the lab, which are these tiny little lab organisms where you can ask interesting questions about using experimental evolution of how evolving really fast can impact their ecological dynamics. I'm going to talk today about my work at Notre Dame, which was conducted at the Smithsonian in Maryland. And then I just started my postdoc in April at Utah State, and I currently live in North Carolina. I'm working remotely, forecasting cheatgrass invasion using a similar ecoevolutionary framework. And while I have been in North Carolina, I've also added a new skill to my CV, which is learning how to drive a tractor. So I come to you from the mountains and I'm coming back to the sea. This is right half an hour away from where I live. And these are really pristine mountain ecosystems. Something I really like about living in Western North Carolina is that sometimes we feel like we can't even feel the impacts of climate change. We're in this like refugia. You're sure things are getting a little bit warmer, but it's not really that dramatic. But when I come back here, it's a big reminder. So again, this looks like a really pristine ecosystem. This is a marsh out of the Smithsonian Environmental Research Center in Edgewater, Maryland. But these ecosystems are really heavily threatened due to sea level rise and a lot of other anthropogenic changes that I'm sure a lot of you are familiar with, given that you live in this area. So hopefully I don't have to make a big argument to y'all, because you do live so close to these coasts, that predicting the fate of coastal wetlands is a really high priority in the context of climate change. These coastal wetlands have a lot of important ecosystem services. At a very basic level, they're a natural barrier between land and water. They provide important habitat for a lot of organisms, and they store a lot of carbon relative to other ecosystems in their soils. And often when we're thinking about predicting the fate of ecosystems, we maybe will study at the ecosystem scale. But something I'm going to challenge you to think about today in this talk is how things from the individual to population to community to ecosystem level interact with each other, and how ignoring different scales, you'll kind of miss big things that are going on in the ecosystem. So you'll notice here that I've highlighted plants. I am a plant biologist and ecologist, mostly by training. But I do want to notice or mention that predicting the fate of coastal wetlands has a lot to do with things other than plant biology. I'm going to talk a lot about plant biology and ecology today, but if we're going to really predict the fate of coastal wetlands into the future, we're going to need to know a lot more than that. So for example, we're going to need to know a lot about sea level rise, what it's going to look like, what its variation and uncertainty is going to be in the future. These coastal wetlands are not, living in isolation. They are right up against some anthropogenic forces like these houses, these hardscapes. Coastal wetlands can encroach horizontally into forests. This is what's called a ghost forest, where marshes, due to sea level rise, kind of move into these forests, changing their ecosystem dynamics. And so this is a lot more than just plant biology, but I do want to talk a lot about plants today. And so what are plants doing in these ecosystems? They provide really important ecosystem processes. And I really like this figure here by Matt Kerwin and Pat McGonigal that shows what these processes are. So above ground in coastal marshes, plants are slowing down water as it comes in and out. It's a tidal system. And that slowing down of water allows sediment to fall out of the water column. And that sediment creates a little layer and builds elevation little by little on top of the marsh surface. So that's that elevation gain via surface accretion. Below ground, plant organs like roots and rhizomes grow into the marsh soil, expanding the volume of the marsh soil. And through that expansion, elevation goes up little by little as well. So that's that elevation gain via subsurface expansion. And it's through these two processes that marshes have been able to keep pace with sea level rise for millennia, building their elevation little by little as sea level continues to rise. What we don't know now is, given accelerated sea level rise, will these plants be able to keep up, and thus will these marshes be able to keep up? Also below ground, what's going on is these plant organs are growing into highly anoxic, highly anoxic soils, meaning they're really low in oxygen. So a lot of the roots and rhizomes are not being decomposed, and that's why marshes are really, really good at storing carbon. So plants are having these impact on ecosystem processes, its accretion and carbon accumulation, things that ecosystem ecologists are really interested about. And they have to make choices about how they allocate their energy at an individual or population level, of how much to put above ground and how much to put below ground. And you could think that those choices that those plants are making might have an impact on these ecosystem processes. So one plant that I study and what I'll be talking about a lot today is Chenoplectus americanus. It's called chairmaker's bulrush. It's a really common sedge in the Chesapeake Bay around here. And yeah, that'll be mostly what I'm talking about today. So these processes that I just showed you in a kind of cartoon format have been formalized into mathematical models to show how plant traits, sea level rise, and marsh accretion or changes in elevation change over time. And so this is another kind of cartoon schematic of how that process works. Plants grow in response to sea level or flooding, such that above ground biomass, or kind of how much is above ground, or how much of the plant productivity is above ground, is maximized at intermediate levels of flooding. If a plant is flooded too much, you can imagine it might drown. If it's not flooded enough, it also doesn't do very well because they're adapted to these tidal systems. So our above ground biomass production responds to flooding. We just talked previously about how above ground and below ground can impact this process of accretion through sediment falling on the marsh surface, and then also the subsurface expansion below ground. So knowing our above ground biomass production and our root to shoot ratio, or how plants are allocating their biomass above and below ground, impacts that. And then if we think, say, on an annual time step, which is like much of these models are, how much the marsh changes in elevation at 1 year will then feed back and impact the amount of flooding the plants will experience the following year, which is then modified by sea level rise, an external driver. And these models have been really useful in making predictions and have been used since the early 2000s. But there are several assumptions in these type of models that might lead to inaccurate predictions. So first, a lot of these models assume that plant root to shoot ratios, or how they allocate above and below ground, is constant across all environments. And part of my dissertation work looked at this. So I went out on the property at the Smithsonian Environmental Research Center, and I captured above ground biomass at the meter level, and then also took cores of below ground biomass. If biomass allocation is constant across environments or across space, we should see a positive relationship here and not a lot of noise. We see the total opposite of that. There's kind of a negative relationship here. There's a ton of noise. So there's not this really consistent biomass allocation across environments. So that assumption might not fly. Another assumption that these models have is that plant traits, except for above ground biomass, do not change over time. So in the previous slide, I talked about how above ground biomass can shift in response to flooding, but none of the other plant traits that are in these models change over time. And that could be problematic. Because in ecology and evolution, we know that there are ways that traits can change over time. So first, we can imagine that plant traits could change over time due to phenotypic plasticity. Maybe these plants are just responding to some change in their environment. So on the x-axis here, I have a global change factor. We can think of sea level rise, elevated CO2, changes in salinity, and we might have some directional relationship with a trait such that as this global change factor changes over time, the trait changes over time. We can also imagine that these plants might be evolving over time as the environment changes as well. So if we were to grow plants from different time periods in the same condition, they would have different trait values. So they have evolved due to genetic changes over time. But these processes aren't included in these models. We know from a lot of evidence from the Smithsonian that global environmental change has really changed these plant traits over the past few decades, and that these have impacts on ecosystem function. But up to this point, we don't really know if this is a product of phenotypic plasticity, if it's a product of evolution, if it's a product of both. This picture here is showing one of the oldest and longest still running elevated CO2 experiments. So set up by Burt Drake in Edgewater, Maryland in 1987. So in the 80s was thinking about how elevated CO2 was going to impact our natural plant communities. So when I joined my project that I worked on for my dissertation, we had this kind of new system where we could address, is it evolution? Is it plasticity? How are these plant traits changing over time? And I think this is the coolest part of the project, in that we can resurrect these plants from soil sediment really down far in the soil, so about 1 meter down in the soil. And we can germinate these plants that are dormant for over 100 years. So on the x-axis here, I'm just showing you the probability of germination success. And I'm showing you a bunch of different seeds that we aged using lead-210 dating. We can see that we have seeds from plants over 150 years old. We're not as good at germinating plants further down in the soil core as you might imagine, but we do have plants from a really long period of time ago. So the process of doing this, we extract soil cores, we date sediment using lead-210 dating, then we match up the sediment with the seeds, and then we can germinate out these plants in the lab and clone them and use them in experiments to see exactly how these plant traits have changed over time due to evolution. And what's really striking is that my colleagues, Jen Summers and Mike Blum at the University of Tennessee, found that there are genetic differences between populations of these plants over really small periods of time when we're thinking about evolutionary processes. Generally, we think about evolutionary processes happening really slowly. But the big thing to take away from this genetic clade diagram here is that the ancestral genotypes, so plants that were dated and resurrected from the early 1900s, were genetically distinct from plants that were germinated from the late 1900s, or what we call descendant genotypes. So there has been genetic change over time across a period that is really meaningful for environmental change in this area. So this is about the time that I started the project. So we knew there was genetic change in Chenoplectus, but we really didn't know how that turned into trait change. And then because we know plant traits are so important to ecosystem function, how that trait change then might in turn influence ecosystem function. But I've been thinking about these ideas for a long time, as I mentioned. So from some of my work from Colorado State, I worked with some of my colleagues on putting together this paper about how this might work in kind of a theoretical sense on a think piece. about how organismal evolution can induce these ecoevolutionary feedback loops. So I showed you this figure before, but I'll walk through it in a little more detail. So we can imagine we have some population of an organism like our plant that's responding via selection to a change in the climate. So anything like elevated atmospheric CO2, changes in sea level rise. If evolution in those traits happens at a magnitude big enough, and for traits that are important to ecosystem function, we can see that carbon cycling, a process that's really important for ecosystem ecologists, could be impacted. If that changes at a magnitude big enough, then we could see that elemental pools like atmospheric CO2 could be shifted, and then it could feed back again to that original selection pressure on the population. And so we have this system where we can look at this xenoplectic system. A lot of people have theoretically thought about how ecology and evolution can be linked together. But unfortunately, a lot of this work hasn't been done yet. I feel like there's a lot of room to connect these systems. But there's historically been this separation between evolutionary biology and ecosystem ecology, in part because there's an assumption that these processes occur on different timescales. But what we have evidence to bring to the table here is that these plants are evolving at rates much faster than people would assume, and on similar timescales to important environmental change in coastal wetlands. So today in my talk, I'm going to try and put the pieces together to show how plant trait evolution adds up to ecosystem level change. Okay, so understanding these ecoevolutionary feedbacks in marsh ecosystems requires some models. So I introduced you through this schematic to an ecosystem model. This is the marsh equilibrium model. It explains these relationships between changes in plant traits, sea level rise, and accretion and carbon accumulation. And then we need an evolutionary model as well that we want to link together to really encompass these ecoevolutionary feedbacks. So this is a model developed by Louise Chevin. And what this model does is it allows a population to evolve in response to a sustained environmental change. So we can imagine in our case, this could be sea level rise, atmospheric CO2, something that's changing over time slowly. And it allows this population to have traits that both evolve and respond plastically. So nothing to do with genetics in response to that sustained environmental change. And then it predicts the demographics of the population. Will it grow? Will it go extinct? And kind of what will it look like over time, and how will those traits shift over time? So I'm not going to get much into the math today. I'm happy to talk about it if people are interested afterwards. We put together these three scenarios of this model. So on the far right here, we have our full ecoevolutionary feedback model. We focused in when we built this model on one trait to start with and one environmental driver. So we looked at biomass allocation and we looked at sea level rise. So we allowed biomass allocation, or root to shoot ratio, to respond plastically to flooding or sea level rise and then also evolve. We had a second version of the model that only allowed it to respond plastically to sea level rise. And then we have our base ecosystem model, which is kind of the null understanding of how these ecosystems work. And by setting up these three model scenarios, we're allowing ourselves to address those two common assumptions that I brought up before, that biomass allocation is constant across environments. So we're now allowing that to not be true, and that plant traits do not change over time. And now we're allowing that to not be true. And so looking at these feedbacks requires models, but if we want to eventually make realistic predictions, we want data to inform these models. So we have a ton of data that informs this ecoevolutionary feedback model, a lot of which are from an experiment that I'm going to talk about in a little more detail later. That was conducted at the Smithsonian, where we grew plants in PVC pipes out in the tidal marshes. And the important part of our empirical data and what makes this model so unique is that we have traits and phenotypic data from plants that are really old. And we can start our model from 1920 and we can run it forward into the future. And so that's what we did. We used our trait data from these ancestral genotypes from the early 1900s. We parameterized our model with that, and then we ran it forward till 2020. And then we used our descendant data to validate what the model predictions were making. So what does this actually look like? So we're going to predict root to shoot ratio, our trait of interest, through time, so from 1920 to 2020. And we have our 3 different model scenarios here. So one where our root to shoot ratio stays constant, that's our kind of null ecosystem model. We have one where root to shoot ratio can respond plastically to sea level rise, and one where it can respond via evolution and plasticity. So what we see here, we have a good check that when we keep it constant, it stays constant. Plasticity, we can see that if we allow root to shoot ratios to respond to sea level rise via plasticity, they decline over time. This makes some sense ecologically and biologically for us. When plants are more flooded, they invest less resources in their roots relative to their shoots. So this makes some good sense given that we ran this simulation under sea level rise. But what's really cool is that when we allow evolution to operate as well, it kind of saves root to shoot ratio from dropping so low. And that's really important because we know that how plants allocate their resources above and below ground has important impacts on ecosystem processes. And because this is an explicit feedback model where we have all those processes together, we can plot up those results as well. So on the left, you're seeing the same graph you just saw. On the right, we have the same scenarios, but we're showing differences in accretion rate, so how fast marshes are building elevation, and then also carbon accumulation rate, how fast marshes are accumulating carbon in their soils. And so I don't want to fixate too much on the exact numbers here that we're predicting, because this is in a sense a theoretical model, but we do see kind of some big impacts here. So how we allow root to shoot ratio to shift across time has a big impact and pretty big effect sizes on these ecosystem processes that we're interested in. And then we can do what I was mentioning with our descendant genotype data, so these newer plants, and we can use that as a validation to see how that compared to our model predictions. So on the left, you're seeing the same graph as you've seen before. And on the right, I'm showing you some data from our descendant genotypes where we've grown them at different levels of flooding, where on the right of the x-axis is less flooded, and on the left is more flooded. And all I'm doing here in this plot is taking the root to shoot ratios predicted from each scenario at 2020 and plotting them by these x's on the y-axis. There's 3 different vertical lines here because each of these 3 scenarios ends up with a marsh at a different amount of flooding at the end of the scenario. So what I'm looking for here when I'm validating is I want to see which of these scenarios most matches up with the data that I have. Now, none of these x's are particularly wildly out of the range of the data, but it does seem that our plasticity only scenario really falls very well in line with the data that we have here. So this suggests that if we're looking at root to shoot ratio and sea level rise, plasticity is the mode of trait change. But I would hazard that this is not suggesting that evolution is not important over time. Remember, we're only looking at one trait and one global change driver. I mean, we know that plants respond to interacting global change drivers, and traits can co-vary, and this can kind of change the results of our study. So this is a good start, but one of my goals is to kind of build this out into more multivariate space. So I mentioned that making kind of realistic predictions using that model takes some data, but if we go kind of to an empiricist side of things, data are just useful for answering some fundamental questions in ecology. And in particular, in my case, how plants will respond to environmental change. So now I'm going to talk a little bit more about that experiment that I showed you a picture from. This is again at the Smithsonian. This is a big experiment we did in 2019 where we grew plants in these PVC pipes in an experimental design, what's called a marsh organ. because the PVC pipes are lined up at different elevation, and it looks like a church pipe organ. So because they're in a tidal creek, that allows them to experience different levels of flooding over a growing period. Those open top chambers allow us to manipulate CO2. And we also replicated this at 2 sites that varied in their salinity. So there are a few different ways that plants can respond to environmental change. We talked about a couple of them. So plants can evolve rapidly in response to change. They also might just shift their traits via plasticity. And it would be nice if it was that simple. But unfortunately, things are really complex in nature. The effect of one global change factor might depend on another, what I call conditional phenotypic plasticity. So the effect of sea level rise might depend on the level of CO2 in the atmosphere. Also complex is the evolution of phenotypic plasticity. So how sensitive plants are to some global change factor may have changed across time. So what I'm showing you here is a line for our descendant cohorts, and then a line for our ancestral plants, and showing that maybe those slopes have changed across time. Maybe plants are more or less sensitive to a driver. Maybe it has a totally different functional relationship. This is what's known as evolution of plasticity. And in theoretical evolutionary literature, this is predicted to be something that happens a lot, but it's really difficult to show empirically. But our system can really take advantage of the fact that we have both these ancestral and descendant plants, and within one experiment, we can see how sensitive they are to global change factors. So this is how we did this in our experiment. So we looked to see kind of all the different ways that these plant traits could shift in response to environmental variation and evolutionary change. So we have plants that were extracted from soil cores from 2 locations. This is kind of the process of extracting the seeds, germinating them, and getting them ready for our mesocosm experiments. Then we exposed them to 3 different global change drivers. So sea level rise by having the PVC pots at different elevations, those 2 different levels of CO2 that was circulating in those open top chambers. And then we replicated this across 2 sites that varied in their salinity. And what we found is that plant trait response to environmental change, which is maybe not too surprising, is incredibly complex. And we found that the evolution of phenotypic plasticity was a really common response, indicating that populations are responding differently to global change factors over time. I don't know how many grad students are in the audience, but we found a significant 4-way interaction, which was totally terrifying and made me want to pull my hair out. But we were able to break it down in a way I think that makes some good ecological sense. So what I'm showing here is above ground biomass is our trait in response to a bunch of different ecoevolutionary drivers. So on the x-axis here is our flooding metric, so how much the plants were flooded over the course of the experiment. On the left is less flooded, on the right is more flooded. The horizontal panels are 2 different sites where the plants were grown at, the brackish and the freshwater site. On the vertical panels are our 2 different aged cohorts, our ancestral and our descendant. And then the colors here represent our elevated and ambient CO2 treatments. So what we can see here is that there is a stimulating effect of elevated CO2. So this dark green is the elevated, the light green is the ambient, but it's highly conditional. It's only at the freshwater site for descendant cohorts, and it's only at the brackish site for ancestral cohorts. And so why could this be? So we know in plant ecology that elevated CO2 can stimulate biomass production under abiotic stress. So maybe that's happening when plants are particularly flooded. So that's why it's only happening in the flooded conditions here. And maybe it's happening when plants are in a salinity condition that they're not used to. So we know that salinity has increased in the Chesapeake Bay over the same time horizon as our ancestral and descendant genotypes across the past century, such that ancestral genotypes historically experienced a lower salinity, and descendant genotypes historically experience a higher salinity. So maybe the plants in the descendant cohort or in the freshwater site are in a salinity they're not used to, need that elevated CO2 to help them out a little bit. And a similar idea for the ancestral cohort in the brackish site. And across a bunch of traits that we measured, we found these complex relationships. So what I'm showing here on the rows are all the different traits we measure, and on the columns are all these different drivers that we have. So eco is that plasticity, eco is that conditional plasticity, evo is evolution, eco-evo is the evolution of plasticity, and then we also looked for gene by environment interactions. You can see that eco-evo, this evolution of plasticity that is normally really difficult to show empirically, is really common. We found a significant eco-evo interaction across 5 of the 7 traits that we measured. What you'll also notice is that this eco column is lit up fully. And this is to be expected. So we're exposing these plants to environments that we know are going to elicit some response based on previous experiments that have been done at Smithsonian. So we did it for sea level rise that we knew there was going to be a response, CO2. So this isn't a huge finding, but the fact that evolution of plasticity was almost as common of a response was. So we now have a framework for modeling these feedbacks and have data that can inform these type of models, but this really only matters if we have this happening at a magnitude big enough for us to kind of change our beliefs of what's going on in the world. So do we need to incorporate evolutionary processes into our models? What kind of magnitudes are we talking about here when we're making carbon budgets for the wetlands or globally? Like, do we need to consider evolution? And so in the last bit of my talk here, I'm going to talk about how we kind of put all of these together to get a magnitude on this. And I just want to note that this effort and really all of the work that I'm talking about today requires a uniquely interdisciplinary team. I'm working with people from computational biogeochemists to genomicists, and really this work wouldn't be possible without all of them. Okay, so quantifying the impact of plant trait evolution on ecosystem change. So how did we do this? We went out into the same environment and we grew plants at a consistent level of flooding, and we didn't throw any other environmental variables at it. So that's in evolutionary ecology, what's called a common garden experiment. So we had a bunch of different genotypes of plants from ancestral and descendant cohorts, and then we looked at how their traits varied. And because the environment is kept constant, we can infer that variation in plant traits is mostly due to differences in their genetic variation. We then used information from those plant traits and scaled trait variation up to ecosystem processes using a version of this marsh equilibrium model that I introduced further. That's a little bit more complex than the other one I mentioned. And what we found when we looked at just the traits alone was that heritable variation or variation in traits due to genetic sources was made up a huge proportion of the total observed variation in the traits. So what I'm showing you here is all of the different traits we measured on the rows, and then on in those colored blocks, I'm using a variance decomposition method to show where variation in traits is coming from. Is it coming from the genotype? Is it coming from the provenance, which means where the soil core was originally from, where we got those genotypes or seeds before we resurrected them? The dark blue is cohort, so are we in the ancestral or descendant age cohort? And those 3 blue categories are all related to genetics or heritable variation. We also have an other category, which is variation due to experimental setup, like spatial blocking, and then residual variation. So across all of our traits, heritable variation explained 15 to 50% of the variation in our traits. And this is particularly true for below ground traits. As you can see in these 3 bottom rows. And this was really exciting because I spent many hours sieving roots. And one of the coolest findings that we found is how plants were shifting their below ground biomass over time. So this is a complex graph, but I'll walk you through it. So the big panel of the graph is showing the cumulative probability of root biomass as you go from the top of the surface of the marsh down. So each one of these lines that looks like an exponential curve is kind of mapping one of those replicate pots. And you're going from the top and you're accumulating biomass from right to left from 0 to 100%. So you're accumulating your biomass and going from 0 at the top of the marsh surface all the way to 100% at the bottom. This inset graph is showing the parameter that describes this kind of exponential decline. And I'm showing this for these two provenances, those two locations where we got the course, and then across the two age cohorts. So what we're seeing here.
Just tell us we have about 5 minutes.
Yep.
Because we got late starts.
Yeah, no worries. Thank you. So what we can see here is we have really consistent changes between ancestral and descendant across our two provenances. And what this is showing is that plants are actually allocating less below ground biomass deeper in the marsh soil over time. So they're shallowing their root profile between ancestral and descendant cohorts. And if we're looking at the root to shoot data as well, they're allocating less biomass below ground too. And we think this is a nitrogen story. So we know that over the course of the past 50 to 100 years, there's been a lot of anthropogenic nitrogen that's been dumped in these marshes. And what plants do when they have a lot of resources is they don't allocate a lot of their own resources into accessing those nutrients. So plants are usually using their roots to get to water and nutrients like nitrogen. So in a case where they have lots of nitrogen, maybe they're putting less resources in those expensive roots and putting more in their shoots. So that means they have a shallower rooting distribution and a lower root to shoot ratio over time. And when we used this experiment to see what are the impacts of this plant trait variation on ecosystem processes, we saw that there were really big impacts. And so what I'm showing you here is those different cohorts and those different provenances and the colored lines. This graph is showing how the marsh elevation changes across time. And this is showing variation in vertical accretion rates and carbon accumulation rates. These individual lines show draws from distributions that account for all of the heritable variation in 3 plant traits in this model. And kind of one of the biggest findings here is when we compare between these descendant and ancestral cohorts, vertical accretion rates were 8% higher and carbon accumulation rates were 18% higher for the ancestral versus the descendant genotypes. And these are magnitudes that are really similar to big experiments that are exposing plants to elevated CO2 or different amounts of flooding. So these evolutionary changes are really big and things that ecosystem ecologists would care about. And what's really interesting is that we saw that plants evolved to have shallower rooting distributions and less biomass below ground. We know that evolution at the population individual level is an adaptive thing. It should be good for them. But what's happening at the ecosystem level is actually really bad. So having less biomass below ground means you're accreting slower and you're accumulating less carbon. And so that's why it's so important to think about these different scales, because if we were just to look at the ecosystem scale, ignoring what the individual is thinking about, we would have gotten our predictions wrong. So I hope that I have convinced you today that plant trait evolution over decades to centuries can impact marsh ecosystem dynamics. And with that, I will skip that and acknowledge a bunch of people and thank you for your time.
We have time for just a few questions.
Yeah, thank you.
So we do have a class coming in behind.
Sure. Yes.
Okay. I'm curious about, certainly plants can adapt and evolve differently depending on how many genes are actually controlling a particular trait. And so I was intrigued by the fact, that when you look at direct evolutionary effects, you know, I mean, is there some prediction that you could see in the genome of that plant to say, oh, well, yeah, that maybe makes more sense because it's a much simpler change genetically that would need to occur in order for there to be root architecture differences?
Yeah, it's a really good question. I would say there is some really exciting work doing the genome of this plant. So I think we'll have a more direct answer to your question.
Oh, the genome's not done?
It's not done yet, but it's in process. So our collaborators at UTK are working on that right now. But I think to get to answer, so one of the reasons that we chose root-to-shoot ratio is because it's not only an important trait that's important for ecosystems, but we can think of it as being a really important life history trade-off. So I don't know exactly the genetic architecture of what's going on behind the scenes there, but to me, it kind of makes sense as to why plants that would be so driven genetically.
One more question for me?
So if it's really bad for the wetland as a whole with the sea level rise, and these plants are changing, could there then be this massive loss of the population that then either causes maybe a local extinction, but then some kind of even more rapid or drastic evolutionary adaptation?
Yeah, I mean, I think that's a totally reasonable prediction of what could happen. So in the last research I was talking about, we looked at trait changes over time and then predicted that into the future, but we were really looking at trait changes over the past 100 years in response to changes in the environment. The pace at which the environment is changing for these plants now way outpaces what has happened over the past 50 to 100 years. So I would imagine that what they're responding to and how they're responding will likely be different over the next few decades. Thanks.
Well, thank you very much. I think we have to pull it to a close.
Yep.
Very
Thank you.